So I really appreciate the opportunity to be able to speak to you at ESSO 2019, and I also appreciate the opportunity to be invited to Poland. So in the previous sessions, all of you have been exposed to very many different disciplines of science. And I was sitting there, I was thinking, yeah, hyperthermia is like really cool because there are so many disciplines that are important in order to make hyperthermia practical for patients. That is one of the attractive things. It's also one of the difficult things because much of the stuff that was talked about, I really don't understand. So what I'm going to do is I'm going to add even more different disciplines. So I'm going to look at molecules in this sense and what their impact can be on hyperthermia. And so in my lab, the focus is the breast cancer associated protein 2, the BRCA2 protein. And in the first part of my talk, I will tell you how studying the BRCA protein actually has implications for thinking about combination therapies with hyperthermia. And that's reviewing of some older work. In the second part of my talk, I will talk about unpublished work that started with looking at heat effects, hyperthermia effects on the BRCA2 protein, but took us in completely different directions to discover new things that have actually implications for how to treat certain group of cancer patients, how to get them a precision therapy. Which is the nice thing about being a fundamental science department, because we can follow wherever the leads are, wherever we find leads, we can follow those up and find out interesting things. So we focus on genome instability. And we do this because genome instability is a hallmark of all cancers, irrespective of the site of origin of this cancer. And this genome instability is due to a defect of these cancers in the DNA damage response. And because that defect is specific to the tumor cells and not the healthy cells, if you could act on that defect, you have a precision therapy. That defect becomes the Achilles heel of that cancer. And that allows you to hit the cancer cells, if you could act upon that, and not the healthy cells. And so that gives us precision therapy. And we all know we need many more precision therapies than we currently have. Lots of cancer therapies are ineffective. Many patients suffer only the side effects and do not get cured. So by designing more precision therapies, we'll increase the quality of life of those patients because if the precision therapy acts on the cancer, not the healthy cells, we get less side effects. And of course, that improves life expectancy of patients. The challenges are twofold. That is, how do you select patients for the specific therapy? And once you have a therapy, because it's a precision therapy, a personalized therapy, if you will, there are a limited number of patients that are eligible for that therapy. And so in the first part of my talk, I will address this second part. So talking about if you have a precision therapy, how can you make it accessible to more patients? And in the second part of my talk, I will talk about selection methods because we discovered a novel protein that interacts with BRCA2, and that if a cancer has that protein, it has consequences on how you can select that patient for a specific therapy. So first, the specific DNA damage response pathway that we study, where the BRCA2 protein is functional in. It's called homologous recombination. It looks very complicated, but is not. So basically what homologous recombination is that if a DNA molecule in the cell contains a break, then it repairs that break using the undamaged sister chromatid. It's basically how you make a jigsaw puzzle, right? So in the cell there is the example, there's the box, which is the intact sister chromatid, and that is used by the cell to pick up all these pieces, the broken DNA, and put them back as they should. And when you make a jigsaw puzzle, you do it with your hands and your eyes, and the cell does it with a protein called RAD51. And it's a protein that finds the broken DNA and then has the interesting property of scanning the DNA and setting up a joint molecule exactly at the place where this DNA is identical to this DNA. And then that joint molecule can lead to repair of the broken DNA. Now, RAD51 doesn't do that alone. And because I said we focus on BRCA2, I'm sure you can guess that the protein that's required to get RAD51 to the site of damage where it needs to act is the BRCA2 or the BRCA2 protein. And it turns out that homologous recombination is a poster child for precision therapy. And that has to do with the discovery that tumors that are BRCA mutant, that can no longer do homologous recombination, are exquisitely sensitive to a drug called a PARP inhibitor. And so the way that works is that inside cells, 50,000 times per day inside every cell in your body, a single-strand break occurs. Now, that is not such a bad lesion. It's easily repaired, right? And it's repaired by a protein called PARP1. And PARP1 finds these lesions and then modifies proteins around that site, attracts the repair proteins, and takes care of that single-strand break. So now, if you inhibit PARP1, what happens is that this is a replication fork, so DNA is multiplying. And when DNA starts replicating while the PARP is inhibited and the single-strand breaks are not repaired, you will run off this single-strand break and convert it into a double-strand break. And that double-strand break, as I just showed you, could be repaired by homologous recombination. However, in cells of BRCA patients, homologous recombination doesn't work. And so in the tumor cells, homologous recombination is defective, not in the healthy cells of these patients because they still have one of the alleles that is not damaged, that is not inactivated. But in the tumor, the second allele is inactivated, you have no homologous recombination, and therefore the combination of PARP inhibition and the homologous recombination defect specifically in the tumor cells kills those tumor cells and not the healthy cells. And that's an example of synthetic lethality. Now, that's an introduction to why we got into hyperthermia, and that was because of an observation. There are many observations. We heard from Professor Datta this morning of all the different clinical trials that were going on, and here's one example, and this was from a clinical trial out of the Erasmus Medical Center, where we see that if you have cervical cancer, patients that are treated with a combination of radiotherapy and hyperthermia have a much better survival rate. And to this audience, and after all these talks this morning, I don't have to tell you that hyperthermia is used in very many different cancers. And we wondered why this effect was. So what would be the effect of hyperthermia? I'll get to that in a second. And also this slide I can quickly pass over because Hans Creuset this morning told us about all the different ways that we have hyperthermia applicators that can reach tumors at very different sites in the body. Now, hyperthermia, of course, on a biological system has very many different effects, very pleiotropic. So it increases blood flow, it induces heat shock response, it makes membranes more permeable, which means drugs can get in cells easier. It has effects on the immune system. So there's all these different effects that happen when you start locally heating tumors in patients. But because we are focused on the DNA damage response, we were interested in what happens to DNA damage repair when you heat those cells. And this was a collaboration with the people at the Amsterdam Medical Center, specifically a very good Polish student at the time. He's now a group leader at the medical center in Amsterdam, Przemek Krawczyk. And so we worked together on this. And what we reasoned was the reason hyperthermia sensitizes cells to radiation is because it inhibits DNA repair. Specifically, it inhibits homologous recombination. So if it inhibits the natural defense of against the radiation, which is radiation breaks the DNA repair pathways put the DNA back together. So if hyperthermia inhibits that natural defense, then of course the cells become more sensitive when you heat them. And so we tested this, arguing that if we genetically inactivate homologous recombination, then those cells would no longer be further sensitized by the heat, because the heat takes out recombination, but we've already taken it out genetically. And so we did this by inactivating a protein called XRCC3, which acts in between BRCA2 and RAD51. And so if we inactivate that, we inactivate the complete homologous recombination pathway. So here's the actual experiment. It's in the older days, so it's an siRNA experiment where we use siRNAs to downregulate this XRCC3 protein. And we see in cells that are recombination proficient that hyperthermia sensitizes cells to radiation. And when we have inactivated XRCC3, this is the curve at 37 degrees, and we can, at 42 degrees, no longer sensitize those cells to radiation. So that was an argument that hyperthermia, mild hyperthermia that's used in the clinic, one of its effects is inactivating of homologous recombination. And we looked at this, so this is just by survival, but we also looked at this in a functional manner by using an assay that actually measures homologous recombination. And that is by measuring gene targeting. So if you have a cell, this is a chromosome, and you transfect that cell with a piece of DNA, what often happens is that piece of DNA integrates randomly anywhere in the genome. So let's say this piece of DNA integrates into a chromosome that's not related to that piece of DNA. That happens most of the time in mammalian cells when you bring DNA in. However, at 1 in 10,000 events in mammalian cells, if you bring this piece of DNA in, it integrates exactly at the place where it's homologous to the mammalian chromosome. So that is called gene targeting or targeted integration, and that's mediated by this process that I just showed you, homologous recombination. So we developed this assay that is basically a FACS assay. So if you bring this piece of DNA in and it integrates randomly, you do not activate this green fluorescent protein, the GFP protein, but if it integrates homologously, it now activates the green fluorescent protein gene, and the cell starts to glow green, which means you can separate the dark cells from the green cells, the fluorescent cells, by a FACS analysis. And so if you do this in mouse embryonic stem cells where homologous recombination is really efficient, if you do this at 37 degrees or at 42 degrees, recombination is about the same. But if you heat the cells for an hour at 41 degrees, you can see that these are the dark cells, so these are the randomly integrated cells, and these are the green cells, the homologously integrated cells. There are much less of those. So also functionally, the heat inhibits the homologous recombination. And then we wanted to get some idea of, yeah, okay, so homologous recombination inhibited, but what is the target? What is the target that the heat goes after? Why is it inactivated? And I already introduced the BRCA2 protein and the RAD51 protein, but there are proteins that work upstream of this. Their names are not so important. They're called MR11 and RPA. They are required in processing these breaks so that BRCA2 and RAD51 can work. So we use cell biological assay to induce DNA damage and then stain for these proteins and see where they are. See whether they are at the DNA damage site, yes or no. And so this was done by a technique we also worked out a number of years earlier together with Prema Kravchek, where we were able to introduce a line of DNA damage inside cells, a line of double-strand breaks, basically by shooting an alpha particle through the cell. The breaks are stained in red, and the repair protein is stained in green, so it co-localizes, it becomes yellow. And at 37 degrees, we see that this MR11 protein is at the breaks, the RPA protein is at the breaks, the RAD51 protein is at the breaks. But if we incubate the cells at 41, we still see MR11, we still see RPA, but we no longer see RAD51. It does not go to the damage. So that means mild hyperthermia, it does not inhibit the early processing of the DNA, of the damage, but it inhibits the loading of RAD51, that very important protein that finds the homology and then puts the DNA back together again. The RAD51 protein levels are not affected by the heat. However, this phenotype, the fact that RAD51 doesn't localize to the site of damage, is also observed in these cells, in the tumor cells of these BRCA patients. And that's because the BRCA protein is required to chaperone RAD51 to the sites of damage. So the logical next step for us was to now stain for the BRCA2 protein and see where that is. Because that protein is required in between these two. And what we find that at 41 degrees also the BRCA2 protein is no longer at the site of damage. And the reason for that turns out to be that the BRCA2 protein is heat labile. So just that heating for an hour at 41, 42 degrees reduces the amount of that protein in the cells. And that can be tested by a Western blot. So here we see RAD51 protein, 3742. The level is not affected, but the BRCA2 protein is affected. And because this protein is affected, this protein can no longer work. And it is dependent on a garbage disposal system in the cell. So there's a system, the proteasome system, that takes care of unfolded proteins. And we can inhibit that. And if we inhibit that system, the BRCA2 protein is no longer degraded by the heat. So we know it's not a non-specific effect, but it's really starting a genuine biological pathway to degrade this BRCA2 protein. Okay. So that, of course, then raises the interesting possibility in that the rare genetic BRCA patients that come in the clinic that have homologous recombination defect are sensitive to PARP inhibition, but it's only a minor fraction of the patients. But we're now saying, okay, what hyperthermia is doing, it is phenocopying that genetic effect that is present in that rare population of BRCA patients. It's just eliminating the BRCA protein from the outside, irrespective of what the genotype of that patient is. So basically we're saying what hyperthermia can do is locally and on demand create this situation. So then the combination of hyperthermia and PARP inhibition should get us along this path. And that should then be what we call an induced synthetic lethality. It's not that the patient has a genetic predisposition, but it's the heat that phenocopies that effect. So we asked then, okay, if we do hyperthermia, do innate cells that are innately homologous recombination proficient, do they become sensitive to this PARP inhibition? And the answer is yes. So this is an early experiment. We used 2 different cell lines. We used a PARP inhibitor, 2 different PARP inhibitors, very early generation, NU1025 and PG34. And in both cases, cells become sensitive when to PARP inhibition if you combine it with hyperthermia. We then moved on and looked in a xenograft model. And we did this together with the group of Timo ten Hage. And Timo is sitting over there, but his name is almost dropping off the screen. Sorry, Timo, I didn't plan it that way. So because his group is very specialized in animal models and hyperthermia. And so because of them, we were able to do this experiment. And so basically, you can see that if you do no treatment or PARP inhibition alone or hyperthermia alone, that there is no effect on the tumor growth. But if you do the PARP inhibitor plus hyperthermia, we can suppress tumor growth to a significant event. So that says it's not only observed in the plastic tissue culture dish, but it's also observed in an animal model with a xenograft for a melanoma tumor in the hind leg of these mice. So far I told you that clinically relevant hyperthermia degrades BRCA2, it inhibits localization of RAD51 to the damage, and then that has the functional consequence of these cells becoming deficient in homologous recombination. They become sensitive to this precision therapy, which now is not genetic, but locally induced. And we can control growth of tumors in xenograft models. So we have now this method to locally and on-demand induce homologous recombination deficiency using hyperthermia. Let's see, where are we going? Yes, so in the clinic, of course, hyperthermia is often combined with radiation or chemo treatment. So at the cellular level, we looked at if we could get some synergy between those and PARP inhibition. So in this particular experiment, we used HeLa cells, radiation, and cisplatin as the chemotherapy, and we used olaparib. So olaparib is an FDA and EMA approved PARP inhibitor that's now widely used in the clinic. And we can see that olaparib itself is a little bit of a sensitizer to radiation and chemo, hyperthermia by itself also, but the combination at the cellular level, cell survival assays, is synergistic. So with radiation and chemo, their combination is more killing than both of them separate. And of course, the next step would then be, can we actually downregulate BRCA2 in human tumors? And the answer is yes. So from our clinic, we got tumor samples from head and neck patients, from bladder cancer patients, and from cervix patients. And we find that in most of these samples, ex vivo, so the tumor material is biopsied, the fresh biopsy comes to the laboratory, we split it in two, and one part is cultured at 37 degrees, the other at 42, and then we do a Western blot for BRCA2 protein. And in most, if not all cases, so for example in cervix cancer, in all cases, we were able to downregulate BRCA2. So in human tumors, it is possible to do it ex vivo, which of course raises the question, should we start up a clinical trial to do this? And the answer is yes, we have done this, and we're including patients for head and neck and bladder cancer to look at whether hyperthermia and PARP inhibition at least don't give any additional toxicity. Now, this morning we also heard that, so Professor Datta talked about, so why is hyperthermia not more often used in the clinic? What's the problem? Well, one of the problems is it's cumbersome and it takes a lot of time. In a radiation department, the doctors are used to treating patients for a few minutes and then they're done. But hyperthermia takes a long time. So together with Prema Kravchuk and our students, Yongxing and Enzo, who is also here in the audience, who will have a talk later this meeting, I think. Yes, there you are. We are trying to think of ways to improve hyperthermia. And we're doing this using heat shock inhibitors. Heat shock inhibitors are also used in the clinic. And we want to see what the effect is on heat shock inhibitors on hyperthermia. So we want to try to see, can we use heat shock inhibitors to avoid thermotolerance? The fact that if you give a second session, that the patients become tolerant to the heat, that the cells become tolerant to the heat. Maybe we can reduce the heating time, or maybe we can lower the effective temperature. So all these things, if we can reduce thermotolerance, shorter heating time, lower heating time, will help make hyperthermia more easily acceptable in the clinic. So what's the effect of heat shock inhibitors of BRCA2? It turns out that BRCA2 is a client protein of HSP90, so that even in the absence of heat, this is an early heat shock HSP90 inhibitor, 17-DMAG. If you just add 17-DMAG to cells at 37 degrees, you already see a lower level of BRCA2. So BRCA2 is already dependent on this HSP90 protein for its proper folding. But then if you add this heat shock inhibitor and high temperature, you're left with even much less protein. So that says the presence of the heat shock inhibitor may even reduce homologous recombination more than heat alone. And we've tested that in the assay that I introduced earlier. So here you can see these were the data I showed you before. At 41 degrees we have less recombination, but now if on top of this we add this heat shock inhibitor, we find that we can reduce functionally we can reduce homologous recombination even more. Which then begs the question, okay, what if in an animal model, which we did together with the group of Klaas Frank and also at the Academic Medical Center in Amsterdam, in an animal model, what happens if we combine PARP inhibition, hyperthermia, and this heat shock inhibitor? And we find that both the combination treatments have some sort of effect, but the hyperthermia plus the heat shock inhibitor and the olaparib drug, the PARP inhibitor, really kill off the tumor growth. And this is an animal model where we're using a tumor in rats. So we have shown it in mice, and now we have shown it in rats with the addition of the heat shock inhibitor that seems to make the treatment even more effective. So can a heat shock inhibitor affect any of these issues here? Thermotolerance, reducing heat time, and lower temperature. So those last two we haven't done yet. We're working on that. But we have done the first experiments on looking at avoiding thermotolerance. So thermotolerance, that if you heat cells on day 1 at 42 degrees, you can sensitize them to radiation. But if you would heat them again, if you heat them at 42 degrees, don't irradiate them, let them sit, and heat them again at day 2, then they become resistant to the radiation sensitization. So that's shown here. So if you don't heat the cells at day 1, but you heat the cells at day 2, you sensitize the cells to radiation. So this is the curve where the hyperthermia is done only at day 2, so they're sensitized from when there's no hyperthermia. But when you add a heat shock inhibitor, this is a clinically used one, Ganetus-PIP. If you had that heat shock inhibitor, you sensitize the cells even more. So that's logical from the data I showed you before. But now, if you add the heat shock inhibitor, let's see, where are we? Okay, yes. So if you heat at day 2, so if you heat at day 1 at 42 degrees, now this line, the sensitized line, no longer can be sensitized by the radiation. So it's the same as the controls. But in presence of the heat shock inhibitor, they still can be sensitized. So what the heat shock inhibitor does, it takes away the thermotolerance, which means you can maybe institute more cycles of hyperthermia in the clinic and make the treatment of the patients more efficient. So for this part then, basically this is what I told you. And the second part of my talk, I told you that heat shock inhibitors can provide new avenues to optimize radiotherapy, maybe allow it to be more recognized in the clinic because it will become easier. And and maybe because more accepted and hopefully more efficient for patients in curing cancer. So this is the older work that I summarized on hyperthermia. And in the next part of my talk, I will also talk about BRCA2 and hyperthermia. But those experiments took us in a completely different direction. And so we were concentrating on the BRCA2 protein, which we know and love because this protein binds the RAD51 protein that I showed you before, right? And it guides the RAD51 protein to the sites of damage. And there it's involved together with another protein that I need to introduce. It interacts with the PALB-B2 protein to repair these types of damages. And it repairs a whole bunch of different lesions. It's a very versatile repair process. So I emphasized double-strand breaks here in the beginning. It also repairs broken replication forks. And the reason that that's important is because many chemotherapies that are used in the clinic induce stress on DNA replication. And it's stress on DNA replication that causes these type of lesions. So this protein and this pathway is very important in repairing many lesions that are caused by common chemotherapies that are used in the clinic. Not only these, but there's also another very important lesion that it repairs, and those are interstrand DNA crosslinks. So those are also induced by many chemotherapies. Mitomycin C and cisplatin, for example, are inducers of this type of damage. And this type of damage is usually repaired by the interstrand crosslink repair pathway. These two strands of the DNA are covalently attached. And that pathway is called the Fanconi anemia pathway. And this homologous recombination pathway interfaces with Fanconi anemia because that can be seen by the names of the genes involved. So the people that studied homologous recombination identified BRCA2, PELP2, and RAD51. And the people that studied Fanconi anemia identified FANCD1, FANCN, and FANCR. And it turns out BRCA2 is FANCD1, et cetera, et cetera. So those are the same proteins. So they operate in the same pathway. Now, we wanted to understand which other proteins interact with BRCA2. And the reason we started doing this was because we wanted to understand how BRCA2 was degraded by that higher temperature. We figured, okay, so that must mean some proteins must associate with the complex and they must target it for degradation. And we, up until today, we still don't have the answer to that question. However, we started those experiments by trying to ask which other proteins associate with BRCA2. And we found a new one that I'm going to introduce now. So the reason we used the discovery that hyperthermia reduces BRCA2. So then we said, okay, if we immunoprecipitate BRCA2 from cells under conditions of hyperthermia, then there is less BRCA2. And all the proteins that covary with BRCA2 complex, that we can identify that covary with BRCA2 are potential interactors of BRCA2. And so we did that by semi-quantitative mass spec SILEC experiments. In order to do that properly, we made mouse embryonic stem cell lines where the endogenous locus of the BRCA2 gene is modified by adding a GFP to its end. And then basically we can do a pull-down experiment where we pull down on GFP and by mass spec experiments ask which proteins co-immunoprecipitate with this BRCA2 protein. And so this is an experiment where all the proteins that are reduced under those conditions come in this quadrant here. And of course, we see BRCA2 there, and we see PELP2 there. I showed you that was an interactor of BRCA2, and we see RAD51 there. But we see a new protein here, HSF2BP, heat shock factor 2 binding protein. And so we wanted to understand what that protein was doing there. Why is this protein part of the BRCA2 complex? So we did a number of control experiments first. We generated HSF2BP GFP knock-in ES cells, PELP2 GFP knock-in ES cells, some control cells. And if we now do the reverse experiment, where we precipitate using HSF2BP, we find HSF2BP, obviously, and we also find BRCA2. So these are not under hyperthermic conditions now, so the HSF2BP and BRCA2 are apparently associated in a complex irrespective of what temperature we're looking at. And if we do the immunoprecipitation via PELP2, we of course find BRCA2, but we also find HSF2BP. So all these proteins pulling down through different components come down together, arguing that they're complex. And then we wanted to understand, okay, can we also do cell biological experiments that give us indications that these proteins interact together? If they would interact together, they would move about at the same way in those cells. And so the experiments we can do is we developed a method that we can individually track single fluorescent proteins inside mammalian cells. And so the way that this is done, that if you have a protein that has a fluorescent tag, then usually there are so many in the cell that you can't see an individual one. However, if you use a specific kind of microscopy, halo microscopy, the fluorescence will go to only part of the nucleus, and therefore it will not activate all the fluorophores, it will not activate all the fluorescent proteins in your nucleus, but only a few. And if you activate only a few, you can discriminate them from the background and you can keep track of them. So it's like, you know, it's one way to identify one protein inside the nucleus apart from many others. And so here are results of those experiments. In the past, we had extensively characterized BRCA2 GFP cells. So what you're seeing here are individual single BRCA2 proteins moving about in the cells. So they're not associated yet with DNA damage. Those cells are not damaged. But if the cells were damaged, they would be on their way to DNA damage. So they're moving about, and they're moving about in exactly the same manner as fluorescent HSF2BP. And of course, from these movies, you can tell, but we can quantitate this because we can combine this with algorithms that can track single particles of these proteins over time in cells. And here you see two of those single particles in a cell moving about. And we identified a number of different classes, ones that are always immobile at the time we're measuring, ones that are always mobile at the time that we're measuring, and ones that have a stop and go behavior. So they're moving, then they're immobile for a while, for the 200 milliseconds that we're imaging, and then they move about again. And you can make these histograms that tell you the populations of different cells. So the cells that are always immobile, they would be in this part of the histogram, and the particles that are always moving are in this part of the histogram, and the stop and go guys are over here. And so this is a quantification of BRCA2 and HSF2BP. They're very similar. And in the past, we had also shown that if you give radiation to these cells, then you immobilize the BRCA2 particles. And that can be seen here. So now the populations of particles are much more here, so they're much more immobilized. And the same holds true for HSF2BP. So this would argue that inside cells, HSF2BP protein and BRCA2 protein are also moving about in the same complex. Not only are they behaving the same in the absence of DNA damage, but if you give DNA damage, you can see a high local concentration. So these now are structures that contain about 50 of these protein molecules. So if you irradiate cells, BRCA2 forms foci, but also HSF2BP forms these foci. So these are now multiple of these proteins accumulated at a site of damage, presumably at work trying to repair that damage. So we find that that accumulation is restricted to S phase. So this red marker here marks cells that are in S phase. So those are cells that are replicating their DNA. And only in cells that are replicating their DNA, these repair proteins form foci. And that's logical because homologous recombination uses the undamaged sister chromatid. So it can only function in the phase of the cell cycle where the DNA is replicated. The protein forms foci not only upon radiation, but also upon treatment with a cross-linking agent, mitomycin C, and also in response to a PARP inhibitor, so olaparib, for example. So all those sorts of damage that's induced by these components, cross-linking, double-strand breaks by radiation, and interstrand crosslinks, the protein accumulates at. To understand if we're working on something important, we looked at conservation. And it turns out that HSF2BP protein is conserved over 450 million years of evolution from fish to mammals. And its molecular interaction with BRCA2 is also conserved over evolution. So for example, this experiment would show that the mouse and the mouse HSF2BP can interact with human BRCA2, and which will become important for the last few slides of my talk. Human HSF2BP can interact with frog, Xenopus, BRCA2. That's what these experiments show here. So the sequence is conserved, but also their molecular interaction is conserved. Now, 20 years ago, HSF2BP was first discovered, and since then there has not been much published on it. And it was discovered and specified as a testis-specific protein. So a testis-specific protein that happens to interact with the heat shock factor 2, HSF2BP. So we found it the experiments I showed you were in mouse embryonic stem cells. So this would argue that maybe a function of HSF2BP would be in meiosis, right, because it's found in testes. But because it's also found in mouse embryonic stem cells, there may also be a function early in development. So we made HSF2BP knockout mice. So here is Western blot from the testes of these mice. So the knockout mice no longer contain the HSF2BP protein. And it turns out these mice are viable. So they're overtly, when you look at them, they're normal. But they're born in a non-Mendelian ratio, about 50% of what you expect, which would argue that there may be a function of this protein in early development. It turns out the male mice are infertile, not the female mice. So the male mice are infertile, and you can see that if you look at stains of the testes, you can see there are no post-meiotic cells, and if you look closely, there are no meiotic metaphases. So somewhere there's a severe defect in spermatogenesis. And you can see that the weight of the testes is way smaller in the knockout mice than the wild-type mice, and you cannot find any sperm in the knockout mice. But you can look at spermatocytes, and you can look at the location of these recombination proteins, RAD51, for example. And I'm not sure if you could see this, but the point is that early in meiosis, in leptotene, you can see these dots, which are RAD51 foci, sites where RAD51 is localized on the meiotic double-strand breaks, to make sure that you can start creating genetic diversity between the parental chromosomes. And in the HSF2BP knockout mice, that doesn't occur. And the chromosomes also do not synapse. And then in the final stage of meiosis pachytene, you can see these beautiful synapsed chromosomal axes in the wild type, and in the mutant that stage doesn't occur. We can't find those. So there's a role of localizing that RAD51, that protein that's also important in mitotic cells, to sites of damage in meiosis. So because we could only find HSF2BP in those embryonic stem cells, and the only tissue we could find it in the mouse was the testes. If we stain for this protein in other tissues, it's not there. Then we looked in cancer cells, in human cancer cells, and we found HSF2BP protein is actually present in a number of cancer cell lines that we all use. So that argues that this meiosis-specific protein, or early development protein, can be ectopically expressed in cancer cells. And so we looked at databases, RNA expression databases, and we found indeed that, for example, glioma or ovarian cancer, the number of transcripts that can be detected of HSF2BP is increased compared to healthy tissue. So this could be an example of a testis-specific antigen. We also looked at the DNA level, so there are all kinds of databases that allow you to look at alterations in genes, for example, amplifications. And what we find is that the HSF2BP gene can be amplified, for example, in ovarian cancer. In 1 of 2% of all ovarian cancers, the HSF2BP gene is amplified. It's overexpressed. Also for breast cancer. So we wanted to see what's the functional consequence of ectopically overexpressing this HSF2BP protein. So we made a system. So we started by looking at chromosomal aberrations, right? So cancer is driven by chromosomal instability. So what if overexpression of HSF2BP would cause chromosomal instability? So we made a system where we overexpress HSF2BP in HeLa cells, look at chromosomal aberrations. We didn't see much of an effect until we started treating those cells with a little bit of mitomycin C. So that's an interstrand DNA cross-linking component. And then we started seeing massive chromosomal abnormalities. And the abnormalities we see are exactly the characteristic abnormalities that are also seen in Fanconi anemia cells. So they're abnormalities that are seen in cells that can't repair crosslinks. And it turns out that HSF2BP overexpressing cells are sensitive to mitomycin C. This is a control experiment. I won't go into the details. Then they don't turn out to be sensitive to radiation, but they are sensitive to this cross-linking agent. Not only are they sensitive to this interstrand cross-linking agent, they're also sensitive to this PARP inhibition agent. And so this is important. So these cells that overexpress it are sensitive to these two different chemotherapies. This will skip. To make a long story short, we looked at, can we find the molecular determinants of this interaction? And we found that so BRCA2 is a huge protein, more than 3,400 amino acids. And we found that only 68 amino acids are important for the interaction of BRCA2 with HSF2BP. This is important because that's going to be a control later on. And we did the reverse experiment where we looked at HSF2BP and we found one point mutant in HSF2BP, arginine 200 to threonine, that no longer interacts with BRCA2. So that's the two important things to remember. If we take out 68 amino acids from BRCA2, we no longer interact with HSF2BP. If we make one point mutation in HSF2BP, we no longer interact with BRCA2. We did a lot of molecular experiments. I won't go to the details. They are in these slides that show that what I just told you is true. We did some biochemical experiments that show the exact same thing. And we did a control experiment where we used HeLa cells and CRISPR-Cas to just take out those 68 amino acids. So now we have cells that have normal BRCA2, except that they're missing the few amino acids that allow HSF2BP to interact with it. And those cells, if in those cells you overexpress HSF2BP, now the cells are no longer sensitive to mitomycin C or cisplatin or a PARP inhibitor. So that says the only target of HSF2BP that causes these phenotypes is BRCA2. Now, the next set of experiments is a beautiful set of biochemical experiments that allowed us to pinpoint the exact step in cross-link repair where HSF2BP functions. It's experiments using Xenopus laevis extracts, and we put in a plasmid that had a single interstrand crosslink. And I'm not going to go to these experiments. They're beautiful experiments because in interstrand crosslink repair there are very many DNA intermediates. And this biochemical system allows us to test all these different intermediates. It allows us to test all the different molecular steps, all the different DNA intermediates in interstrand crosslink repair, whether they're affected by overexpression of HSF2BP. And by doing this, we came to the conclusion that what happens, so now we're just going to go right through this, because I'm not going to show you all the data, because I'm running late. So it turns out, if you have, in order to repair this interstrand DNA crosslink, at one point, BRCA2 needs to load RAD51 on that substrate. And it turns out that the overexpression of HSF2BP causes the RAD51 protein to be absent at the site where it needs to repair the interstrand DNA crosslink. And here's a control, so if we add the protein that can interact with BRCA2, RAD51 is perfectly well loaded. So we have discovered now that overexpression of HSF2BP causes a specific defect in repair of a damage that's induced by a widely used anti-cancer therapy, namely cisplatin. So this would argue that, and I also showed you that HSF2BP is ectopically overexpressed in numerous cancers. So this would argue that HSF2BP, a protein we discovered because we were interested on the effect of hyperthermia on BRCA2, HSF2BP overexpression in cancer patients can be a marker for guiding these patients along certain therapies, including cisplatin therapy. So even though we started with questions about hyperthermia, we didn't solve that part of the puzzle yet, but we did discover a new marker that we may be using in the clinic to guide patients along interstrand cross-link therapy. And with that, we're at probably the most important slide, which is the acknowledgement slide. I think I showed names of people along the slide as I went along, so I'm not going to mention all these names. But with that, I would like to thank you for your attention and be happy to take your questions. Timo.
So I might be off completely, but one thing I think is striking, to me at least, is that HSF2BP is produced in the testis, which has a lower temperature than the rest of the body. And then it plays a role there. So then it's not produced in normal cell. Has that to do with that there's a higher temperature in these other cells? Is there a relationship with
Actually, that's a very interesting question. And you know, that could be tested, right? We could, we could see if the expression of the protein is actually induced when you lower temperature in cell culture.
Because that would then also play a role in if you have hyperthermia. What happens to the level of that protein then?
Yeah, so when we discovered it and the name was related to heat shock factor, that's the first. So I thought, okay, now we got the protein that is required for BRCA2 to be degraded. And it turns out it's not the case. So if you look in HSF2BP knockout cells, you can still degrade BRCA2 by hyperthermia, and HSF2BP protein is not affected by hyperthermia. So yeah.
Okay. So
too bad.
So now that you know quite a bit about the system, do you think that if you affect that over time cells might overcome it by using other pathways, or do you think this is quite determinant for that process? So do you think that the repair might be, there might be another route rather than BRCA2 when you...
Oh yes, for interstrand crosslinks. So yeah, that is has been one of the most molecularly most mysterious pathways of repair, because it's the most complicated, and people are now starting to discover also other pathways of interstrand crosslink repair that can be used that don't depend on the BRCA protein. So yeah. Yes, there's a question over there. Oh, sorry.
If hyperthermia can inhibit BRCA1 or 2, can it be mutagenic?
Yeah, I mean, the thing about hyperthermia, the nice thing is that in order for it to be mutagenic, you probably have to have a sustained longer period, right? So because in the clinic, it's only given for a very short period of time, and therefore I expect that the misrepair that could happen during that period of time will not substantially contribute to mutagenesis.
Yes, my question would be if you expect that other platinum derivatives like carboplatin or oxaliplatin will act in a similar way, or if this might be something completely different.
In general, they act in a similar way. So the advantage of carboplatin, for example, is that it's less. So the reason to use carboplatin in the clinic, as far as I understand, is it's less reactive with proteins. So its effective concentration may be higher. But both these platinum derivatives work through similar pathways as cisplatin.
If I understand correctly, you're saying that the hyperthermia can make the personalized medicine of cisplatin available to many more breast cancer patients.
PARP inhibition, yes.
If that's the case, then two things. Is there a clinical trial on the way? And,
yeah,
and secondly, what's this going to do to the cost of the treatment?
Yeah. So this is one of the frustrating things, right? So mechanistically, we understand, and we suggested that, okay, hyperthermia should be combined with PARP inhibition, right? Okay, so let's go do a clinical trial. So we connected up with AstraZeneca for olaparib. We wrote a protocol for head and neck and bladder cancer. AstraZeneca's olaparib are pills, and they're huge. And it turns out head and neck patients can't swallow them. So we're not accruing patients in the head and neck arm of the trial. And for the bladder cancer, somehow clinicians are reluctant. So to keep, they rather have more standard treatments for these patients. So we're having a hard time accruing patients in those trials. So as Professor Datta said this morning, we need to, as a single site, it's really hard to get enough patients in those trials. But we're trying, and but it's not moving fast enough for my taste. All right. Thank you very much. Oh, one more question. Okay.
So you want substitute, you can combine a PARP inhibitor in BRCA deficient cells, and you want add in other cells hyperthermia. But it is a large difference if a cell is BRCA deficient or you add hyperthermia, which is only working for some hours.
Right.
How long and how many sessions you would recommend to substitute BRCA deficiency?
Yeah, so we have looked at that, so that would depend on the resynthesis rate of the protein, and so so we've looked at this only in cell lines, and it seemed to be a little bit dependent on the cell lines, but in general, it's about 6 hours that it takes for the protein to come back. So you have a window. So the difference with genetic BRCA mutants, of course, is that the tumor is always BRCA deficient, and with hyperthermia, it's only going to be BRCA deficient for a relatively short period of time, maybe six hours or so like that. But clearly, on the cellular survival experiments and the animal experiments, it was enough to make an effect. But yeah, in the end, in the clinic, we would have to test it.
And would you repeat it often? How often?
Yes. So you would. So basically, the clinical protocol is to start the patient 2 days ahead of time on PARP inhibitor, do the hyperthermia, keep it on there for a day still, and then in the next cycle do it again. Yep. Within the 6 cycles of hyperthermia.